All right, in this segment, I want to consider issues of common and rare variation. And once again, and as usual, I'm going to be doing it in the context of SNPs, but the discussion is quite general. So we know from our mutation spectrum, or SNP frequency spectrum, that we can have common variations and rare variations. And let's think about our favorite genes and what this might mean. So common variation is usually defined to be minor allele frequency greater than 1%. That is, more than 1% of any given population has the minor allele in the population. Whereas obviously rare would be a minor allele frequency usually much less than 1%. And then there's a gray area where you can argue about whether it's rare or not. So this is important for a number of practical reasons. And one of the things that's important for is our decision on how to measure SNPs. We typically measure SNPs on SNP chips. And I'm not going to discuss this. I think in other segments you're going to learn about SNP chips. But the key point is these are chips that have about 1 million SNPs on them and can simultaneously measure 1 million alleles of SNPs in the human genome. Now, when they build these SNPs chips, they have two considerations that they have to kind of satisfy. The first consideration is they would like to space them evenly in the genome. So the genome is 3 billion bases. You can put down 1 million SNPs, that is 1 million locations in the genome. It's not going to be a full genome sequence. It's just going to be assaying 1 million locations. So if you get to pick 1 million locations, and if you want to space them evenly throughout 3 billion bases, that means you should put a SNP every 3,000 or so bases. So that's going to be the spacing of the SNPs on average, every 3,000 bases. That's cool. That's pretty high dense sampling. And you'll have the whole genome covered. You don't worry too much about covering every base of the genome. You might worry a little bit. But remember, because of LD, a topic we've spent a lot of time talking about, because of linkage disequilibrium, these bases will be correlated with bases that are near them. And so you're measuring them, but you might be able to detect signal from other bases in the region. That's the first consideration. The second consideration is you want to measure SNPs that will be different in different people, which means you don't really want to measure super rare SNPs. If you measure super rare SNPs, most people will have the common allele. And you will have boring SNP chip results where you've spent a fair amount of money. By the way, these cost about $200. But you're doing these studies on tens of thousands of people, so it adds up. But the point is, if you're doing 1 million measurements, you'd like to get variability. And you don't want everybody to look alike. So you also want to pick SNPs that are common, as defined above. They have a minor allele frequency. In this case, at least 1 out of 100 person, for 1%, will have the minor allele. And so you'll get some diversity in your measurements, not everybody having the same SNP. So when they design these SNPs, they go every 3,000 bases, they look around, and they pick the most common SNP in that region. Okay. So there's a couple of things you can discover in the context of pharmacogenomics. You can have, and one of the things that this leads to is there will be an excess of discoveries in common SNPs, not because they're the most important, but because they're the ones that we're measuring most commonly. So we have our favorite gene, let's say a cytochrome P450, one of these cytochrome, and it's a gene. There's the exons and the introns. And it will be sampled every 3,000 bases or so. There will be a SNP. So let's say you take one there, you take one there, you take one there, you take one there. These are the SNPs that are on the chip. Again, common, spaced 3,000 bases. A couple of things to notice. Most of these are not hitting inside the gene. This is in the intron. This is on the intron. This is on an exon. This is in an exon. When we do science, and we're going to be talking about this in another segment, we're looking for correlations between the allele in a SNP and drug response. So let's say this cytochrome is involved in metabolizing a drug. Perhaps this is a TG SNP, and when it's a T, the drug seems to require a high dose in order to get a good effect. Whereas when it's a G, the drug works okay with low dose. That's what we call an association. We've associated the alleles of a SNP with a certain phenotype or outcome that we care about. This is pharmacogenomics. We care about drug-related phenotypes. Now, this is a statistical association. It says that when I see a T, I tend to require a high dose. But when I see a G, I tend to require a low dose. Now, here's the key question. Do we believe that that specific location in this gene is causing this requirement? And the answer is, in general, no. Because this SNP may be an LD with all kinds of other SNPs to the left and to the right, even past its neighboring SNP. So this is, I'm going to call this my LD fireworks, okay? That's not a technical term. Do not repeat this to anybody. But because of this SNP is in a region of the genome, and because we know LD works, it's possible that there are other SNPs, and I'll draw their positions now, that are the ones that are actually changing the protein to require a high dose or a low dose. This is just really tagging that region. And in fact, in many times, it's called a tag SNP. An important false inference that can be made, and you have to be careful not to make it, is that just because a SNP is strongly associated with some kind of phenotype, that it is the causative SNP, that it is involved in the mechanism by which that drug response is different. More likely, because of the way these SNP chips are made, this is just tagging a region of the genome that needs to be more carefully studied in order to figure out which of the variations in that genome are actually responsible. And so, for example, the ones that are in the middle of a coding region of the gene are very good candidates, because you can imagine them changing the function by changing an amino acid. Another possibility would be SNPs in the control region that control how much of this gene is expressed inside a cell. You can imagine that if you change the expression level, then it might change how much of the drug is required. So this is kind of a discussion that we tend to look for common variants because we want to get good diversity in our data. Those common variants are spaced out evenly. We often can find associations between common variants and phenotypes that we care about, but it doesn't mean that that's the causal variant. Okay, I want to go to one other topic now, which is the effects of selection. In most of our discussions of genetics so far, we've been talking about mutation, recombination, and we were talking about luck. Were you lucky enough to reproduce? Did your mutation wind up being fixed in the population, and did your mutation grow up to be a population? I'm sorry, did your SNP mutation grow up to be a polymorphism? That's all in the case of no selective pressure. But we know from evolution that there's selection. There can be positive selection, which means a variant, like a SNP, helps the organism survive. And thus tends to increase in frequency because the people who survive have babies. Those babies have the good variant. Those babies do well, they survive more, and so for positive changes in the DNA that add to your fitness to interact with the world, those will tend to go up in frequency. Of course, on the flip side, there is negative selection, where the variant hurts survival. And in general, those will decrease in frequency. Because, again, the offspring have unfavorable mutations. They are less fit to do well in the environment. Maybe they're more prone to infections. Maybe they can't metabolize certain kinds of foods, so they get infections, they get starving, they die. It's very sad. And we don't see those variants. And I want to talk about what the effects of these can be on the genome. So positive selection is very interesting. So and I'm going to take you through a story here of positive selection. So we have a bunch of haplotypes. I'm going to call them H1, H2, H3, H4. And we've talked about haplotypes before. I introduced them. For example, it could be GAT and TAT and TAC, etc. Let's do one more. GAC. ... A lot of haplotypes floating around in the genome. And let's say that this distance is pretty large. This is a large distance. So there's lots of recombination. I'll put a little note here. Lots of recombination happening. And if you will, randomizing these associations and eroding LD. Okay, so we're sitting there. That's fine. But we're sitting, let's say this is Africa, and there's malaria in Africa, and this malaria is a terrible drain on the population, and it's killing lots of people. It's killing all the different haplotypes, except at one point, somebody gets a mutation. Let's say it's on TAT background. Okay, so there's a TAT, and now there's a new mutation here. It's a G at a new position. Maybe it was always C, 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 and now we have a new G. And this confers malaria resistance. So let's think about what's going to happen. This person with this haplotype has just won the lottery. They are going to survive. Their children are going to survive. Their grandchildren will survive, because malaria is the biggest killer by construction. I kind of hypothesized that. All these people are going to have trouble. Their offspring are going to die. And if this is sufficiently favorable over tens or hundreds of generations, this will start to dominate the population. And people who inherit at least this segment of the chromosome will have a huge advantage against malaria. What this means is that all the LD erosion that happened over here kind of gets erased, because now this is the winner. And so he is a new haplotype that in only 10 or 20 generations has kind of taken over the population, at least this segment of the chromosome. The rest of the chromosomes, not so much. Which means the diversity and erosion in these haplotypes have basically been lost. This one dies out, this one dies out, this dies out, this dies out. And you're getting a restart on LD erosion. I'm going to say LD erosion starts anew. Why am I telling you all this? Well, it's very interesting because you can detect these positive selection variations, because whereas the average distance of LD will have eroded, so just to draw it here, let's say that after all of this, before this mutation happened, the average distance of LD was like these little boxes. These are called LD blocks. LD blocks. And they're blocks within which LD is still detectable. And they get smaller and smaller as you have recombination and randomization. The LD block is essentially reset here to be one big block. So when you look at the genome for signs of positive selection, this is what you see. This is our chromosome. You're going to see little tiny LD blocks, small, 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 and then you might see a big one, and then small, 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 and then a kind of big one, small, 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 big. That means there's been 3 times in the history of this clan of humans where a positive mutation has reset the erosion clock, and you now have much bigger blocks of LD. And then surrounded by the small blocks that were not selected for. Again, over time, there will be recombination. This will erode, this will erode, this will erode. But where this may have been happening for 100,000 years, if this only arose 20,000 years ago, that block will be much bigger. So we can use this to detect signs of positive selection in the population. Similarly, for negative selection, it's a different phenomenon because you will not have these big blocks which are good. Instead, you will have the elimination of bad blocks. It's kind of the flip side of the same coin. Less of interest is the negative selection because we just know it's deleterious and we don't really see it because it gets selected out. We do see, however, the signs of positive selection.